Today I'm answering an IGCSE Edexcel biology paper, and it's the June 22 paper, 1B. Diving straight in at question 1. The human body has different hormones that are produced by endocrine glands. The diagram labels some of these endocrine glands in the body. Which gland produces insulin? So that is the pancreas, which is the vine-shaped gland. Vine leaf-shaped glands, so that's A. Do make sure you can label the other ones, just as a matter of course. Because this is on top of the kidneys, and renal means kidneys, this is the adrenal gland. We've got the ovary, and then testes down here. So do double check that you can do this. Which gland produces progesterone? Progesterone, remember, is responsible for maintaining the uterus lining. It's made by the corpus luteum, which is in the ovary, so that's why the answer here is D. The human body has two types of system of communication, nervous and hormonal. Students research the speed of the nervous and hormonal communication. They find this data. Hormones travel at a speed of 420 centimeters per minute. Nerve impulses travel at a speed of 55 meters per second. Why have they chosen different units? Determine the ratio of the speed of nervous communication to the speed of hormonal communication. Give your answer in the form n/1. Okay, let's get these units sorted. I'm going to get 420 centimeters per minute into seconds. So that becomes 4.2 meters per minute. And then I need to divide 4.2 by 60 to get it per second. Lyra, please go away. No, don't stand on me when I'm recording. Kitten. Okay, so at least we're in the same unit. So then it becomes 0.7 to 55. And to get it into the right ratio, we need to divide both by the smallest number. So it becomes 1 to do 55 divided by 0.07. So it becomes, just to do it exactly as they've done it, 786 to 1. Describe 3 other differences between the nervous system and the hormonal system. So the nervous system uses neurons. Make a full comparison. The hormonal system uses blood. The nervous system uses electrical impulses. The hormonal system uses chemicals known as hormones. Don't give up, you need to give a third one. Nervous system has a short-term response. Hormonal system has a long-term response. Biologists classify organisms into different groups. One group of organisms is fungi. Complete the passage about fungi. Fungi do not carry out photosynthesis. Their body is usually organized into a mycelium made from thread-like structures called hyphae. Gosh, you're going to have to learn this very carefully, guys. Fungal cell walls are made out of chitin. Fungi feed by extracellular secretion of enzymes onto food material and absorption of the organic products. This is known as saprotrophic. Remember, that is all mentioned in the specification right at the beginning. A student investigates the effect of temperature on the rate of anaerobic respiration in yeast. The student measures the rate of gas produced in centimeters cubed per minute. The graph shows their results. Name the gas produced by yeast during anaerobic respiration. Remember, they produce ethanol and carbon dioxide. Explain the effect that increasing temperature has on the rate of gas production by the yeast. So we're going to have to split our answer up from 15 degrees to 40, and then what happens in this portion of the graph. So from 15 degrees until 40 degrees, gas production increases. Why is that? We're explaining it. Because the enzymes have more kinetic energy. And collisions occur more frequently with substrate molecules. This is really a chemistry collision theory answer. Above 40 degrees, remember what happens to enzymes is they denature. So that means that the substrate no longer fits that deformed active site, which you can say, but it's still the same marks. Describe how the student can measure the rate of gas production in the experiment. So they want to use a gas syringe to find the volume of CO2. But remember, it's a rate, so it's per unit time. So let's say in 1 minute. And that's how you get your second mark. Vital capacity is the maximum volume of air that a person can force out of their lungs in one breath. The graph shows the relationship between vital capacity and the age for a large number of people. The vital capacity plotted is the median value for each age. State what is meant by the term median value. That is the middle value. Give a reason why the median is used rather than the mean. It's actually because it means that the data is less affected by anomalies. Describe the relationship between vital capacity and age shown by the graph. So remember, that's saying what we see on the graph, it doesn't require any explanation. So we can see that the vital capacity increases steeply up until the age of 20 to 25 years. Then it levels off and then decreases steadily. Explain why vital capacity changes with age. You want to kind of think about what's going on inside the thorax, because remember, those intercostal muscles and the diaphragm are responsible in terms of how they contract and how they relax. How they relax will affect the vital capacity, because remember, that's forcing air out of the lungs. So we need to say that the diaphragm muscle is weaker with age. Lower vital capacity above 25 years. And then let's talk about why does it increase up until 25 years? The lungs are growing in size. So sorry about my handwriting. The cat is stopping me from moving my hand properly. Age is not the only variable that can change vital capacity. Give 2 other variables that can affect a person's vital capacity. So actually, amount of kind of sports, gym going will definitely affect a person. So let's call that the fitness, and naughty lifestyle behaviours like smoking is obviously going to reduce vital capacity. Describe a method you could use to demonstrate the effect of exercise on breathing rate in humans. Well, it's only worth 3 marks, so you're going to have to pick these carefully. Don't worry if you write too much. I'll pick out the correct answers. So what's our independent variable? What are we going to change? So some students will exercise. Others will be at rest. What will our dependent variable be? So what are we measuring? Count the number of breaths per minute. What about our control variables? What are we keeping the same? Age of student, gender, fitness, And then, as always, repeat and calculate an average. Scientists collect data from a grassland ecosystem. For each trophic level, they determine the mean number of organisms in a square metre, the mean dry mass of the organisms in a square metre. Draw a labelled pyramid of numbers for this data. So here's our numbers. Make sure you've got everything laid out properly. So remember, we'll start with the producer. Which will have the widest bar because you've got the greatest number. Then we go up to the primary consumer, followed by, can't really fit that in, so I'm going to write it there. Followed by the secondary consumer. And then a very skinny tertiary consumer. Obviously, feel free to write that out in words. Describe how you would collect data to find the mean number of producers per square metre in the ecosystem. Hoping this is shouting quadrats. So use a random number generator to randomly place the quadrats. Count the number of producers. Place several quadrats. The mass of organisms at each trophic levels is called biomass. The percentage of biomass in the producers that is transferred to the primary consumers is 4.5%. Calculate the percentage of biomass in the secondary consumers that's transferred to the tertiary consumers. Looking at these two numbers really, so we just need to take our tertiary consumer dry mass and put it over the secondary consumer to give us 23 to 2 significant figures. Comment on the energy transfers in this ecosystem. In your answer, refer to the data from the table and percentage of biomass transferred. Okay, really it's asking why so much energy lost at each stage of the food chain. So let's list all those key points. Energy is lost by respiration, excretion, which remember is removal of metabolic waste, digestion, removal of feces, keeping warm. Not all of the organism is eaten or digestible. So the marks are pointing out that energy is lost. Then there's 2 marks for pointing out ways in which that energy is lost, but that's the maximum they'll give you for that sort of answer. So then we just need to talk about that so much energy is lost through all the different trophic levels that by the time you get to the top of the pyramid, you have very few organisms left. And that's how you'll guarantee your 4 marks. The diagram shows a wind-pollinated flower with some structures labelled W, X and Y. Describe how structures W, X and Y are adapted for wind pollination. So there's our feathery stigma. Stigma is feathery. Anthers are exposed and filaments are long and hanging. And all this means, remember, is that the wind will blow away that pollen and mean that the stigma is more likely to trap it. Structures W, X and Y adapted for wind pollination give 2 other differences between wind-pollinated flowers and insect-pollinated flowers. So the insect-pollinated flowers need to attract their insects. So they do that by having a nectary, and a strong scent and large, colourful, flag-like petals. Wind-pollinated flowers often cause an allergic response in people. This is known as hay fever. Most people in the UK who get hay fever have symptoms from April to September. That's a long time. The graph shows the changes in total pollen count for 3 different plant types from March to September. As part of an investigation into pollen allergy, 5 people keep a diary of their hay fever symptoms. They do this for the same year as the pollen count. Use the data in the table and the information from the graph. Discuss the likely causes of the allergic responses in each person. Oh gosh, this is such a tedious question. Right, let's look at person A. So they've got severe symptoms in April and May. So that's here. So we can see that their hay fever is being caused by trees. What about person B? June, July. They've got 2 types of hay fever going on. That's weed and grass. Slightly more mild on the weed allergy. Person C, April to September. Gosh, that's an awful lot of allergies. So they're allergic to all pollen types if we use that key. Person D, they are not allergic at all. Person E, June to September. So they're mainly allergic to grass and weed. What is meant by the term immune system? This is when white blood cells, e.g. phagocytes and lymphocytes, destroy microorganisms known as pathogens because they cause disease. The diagram shows a plant cell in distilled water and a plant cell in concentrated solution of salt. This is already screaming osmosis, which is the structure labelled M, so it's the very outermost part of the cell, it's the cell wall. Which structure is labelled N? It's the cytoplasm. Give the name of the liquid found in the gap labelled O in the cell in the concentrated solution of sodium chloride. Because plasmolysis has occurred here, you can see how that cell membrane is pulled away, which means it's opened up a gap that allows that sodium chloride solution to flood in. Explain the differences in the appearance of the cell in distilled water and the one in sodium chloride. So we really need an answer to do with osmosis here. So in distilled water, water has entered the cell. Just have a look, it should look really swollen. Yeah. By osmosis from an area of high water potential. You can write that out in full or just write the sign, which is the surrounding solution. To an area of lower water potential, which is inside the cell. The cell is turgid. Now the opposite has occurred in the sodium chloride solution. So I'm going to steal that wording from higher up. And I'll change colour and just show how you can change the wording. So in sodium chloride, water has left the cell by osmosis from an area of high water potential, which remember will be inside the cell, to an area of lower water potential, which will be that sodium chloride solution. I've written the chemical symbol there for sodium chloride. The cell is flaccid or plasmolysed. Describe an experiment you could do to show how different concentrations of sodium chloride solution affect the appearance of plant cells. So do you want to talk about potatoes or do you want to talk about onion skin? For some reason, you can't score full marks if you do the potato one. You can only get a maximum of 3, so we'll have to dive in with the onion skin one. Use red onion cells. What's our independent variable? So what are we changing? The concentrations of sodium chloride solution, e.g. 0 molar, 1 molar, 2, 3, 4. What's our dependent variable? What are we measuring? What are we examining? It's a bit weird, but because we're not really measuring anything, but we're going to observe the onion cells under a microscope, just because it talks about the appearance. Give a time frame. After 5 hours, just pick something sensible. And let's see how many marks we've got already. We've got a mark for saying using the red onion cells. We've got a mark for talking about that we want to choose different concentrations. We're leaving it for 5 hours, that's a mark, and we've said our dependent variable, which is observing the onion cells. So that's actually 4 marks. The diagram shows the human heart with 4 chambers. Which blood vessel brings deoxygenated blood to the heart? So remember, let's just have a look at blood flow. This is coming from the lungs. That's going to the body, and it is oxygenated because it is the left side. Now, if we look at the right-hand side, remember that blood is deoxygenated. It's coming from the body where the cells have used up the oxygen in their respiration. It's going to the lungs to become oxygenated. It's worth labelling these things to get your eye in. Which blood vessel brings deoxygenated blood to the heart? It's this one here. It's U, the vena cava. Which chamber pumps oxygenated blood away from the heart? So it's leaving here via the aorta, so the chamber responsible is the left ventricle, down here Z. Explain the difference in the wall of chamber S and chamber Z. So we're comparing the thickness here. So Z is the left ventricle, which is a thicker muscular wall, as it must pump the blood at much higher pressure and much further. All the way around the body. You could have obviously given the opposite argument for S. Humans need a balanced diet for healthy growth and development. Give the function of 3 different components of balanced diet. Finally, a straightforward question. So, carbohydrate is needed for energy. Do you want to talk about proteins, fats, vitamins? Fats are needed for insulation. Proteins are for the growth and repair of muscles. Scientists investigated the link between body mass and coronary heart disease in Australia. The scientists recorded the number of heart attacks in a population of 850 people over 20 years. They classified people as normal mass, overweight and obese. They calculated rates of heart attacks that allowed a valid comparison to be made. Evaluate what the data shows about the relationship between classification of body mass, age and heart attacks. Okay, let's look at body mass. So as body mass increases, we can see across all age groups, the rate of heart attack increases. What about age? So again, we can see that that also increases, apart from for this obese category over here. Let's talk about the quality of the study. The study is good as it runs over 20 years and involves a large number of people. So there's a further 2 marks. What's bad about the study? But only people in Australia are included. And they don't include any underweight people either. Fur colour in rats is controlled by a gene with 2 alleles. One allele codes for black fur, the other one codes for agouti. Several female rats with agouti coloured fur are mated with several male rats with black. All the offspring have agouti coloured fur. Explain which allele is dominant. Agouti, as all offspring have this coat. A male and female rat from these offspring are then mated together in a second cross. I'm just going to use R to represent the fact that they must have one agouti allele, which is the uppercase R, and one black allele, which is the lowercase r. Some of the offspring of this second cross have agouti coloured fur and some have black. Yep, I would expect that. Draw a genetic diagram to show this second cross. Include the genotypes, gametes, and phenotypes. Right, do set out your answer like this, because then you're going to be guaranteed that it's correct. So we're going to make a small table here, which is the phenotype, genotype, gametes. Doesn't really matter with this because we know that they were all agouti, that first cross. I've already assigned R's. So that means the mum's eggs look like this. The dad's sperms look like this. Then we're ready to do a Punnett square. So we have 25% are black furred because they have that genotype. 50% are agouti with this genotype. And so in total 75% are agouti but with this genotype. So really it's 3-to-1 agouti to black fur. Calculate the probability of any one offspring from the second cross being male with agouti coloured fur. So there's a 0.5 chance of them being male. There's a 75% chance of them having agouti coloured fur. Look. Pop that into your calculator to get 0.375. Scientists observed that genes that control fur colour in rats can affect rat behaviour, such as how tame they are. Other genes that control the size of the adrenal glands and the production of neurotransmitters also affect rat behaviour. Scientists also noticed that coat colour is associated with differences in anatomy and physiology, such as the size of the adrenal glands and the production of neurotransmitters. State the name of the type of genetic control where many genes control one phenotype. The key term there is many genes. Many is given by the word poly. Genes is genes, so polygenic. Explain why the size of the adrenal glands and the production of neurotransmitters would affect rat behaviour. 3 marks. Adrenal glands, what do they produce? Well, they produce adrenaline. And then let's talk about what adrenaline does, which increases heart rate, breathing rate, dilates pupils, makes fur stand up on end, etc. What about neurotransmitters? if more neurotransmitters are produced, remember they pass a synapse and carry the electrical impulse onto the next neuron. So the electrical impulses will be conducted faster and reflex actions will be quicker. Some rats with white fur also have pink eyes. These rats have pink eyes because they do not have pigment in their irises. This means that the irises let light pass through, unlike the coloured irises found in other rats. Well, that's interesting, look, because if that's your front-on view, there's your pupil, here's your iris. So in normal rats, the light only enters the pupil, but with these albino rats, it enters the whole eye. Explain how this difference in the iris affects vision in the rats with pink eyes. Oops, I keep accidentally answering these questions. Iris allows more light to enter the eye. Which could potentially damage the retina, because remember, the whole point of having a pupil that dilates and constricts is to restrict the light entering the eye when it's too bright, therefore preventing damage to the retina. And if you damage the retina, that could lead to poor vision, because remember, the retina is where the photoreceptors are found. Don't worry about filling all these lines. If you think you've made 3 good points, don't overfill. Selective breeding has been used to develop modern varieties of wheat. Describe how scientists could use selective breeding to increase wheat yield. So, scientists choose, so we're kind of describing the process of selective breeding, wheat with a higher yield. And allow these to self-pollinate. The mark scheme says allow words like mate, but it's not an animal, so try and be sensible with your wording. And the key thing with selective breeding is offspring with higher yields are chosen again and made to self-pollinate. And the process repeats over several generations, and that's the key underpinning feature of selective breeding. During a long-term study of selective breeding, scientists collected data for the mean yield of wheat in tonnes per hectare. Plot a line graph to show how the mean yield changes from 1840 to 2020. Use a ruler to join your points with straight lines. So the year will be the x-axis, because that's what you're changing, the independent variable. What you're measuring is the yield in tonnes, so that's your y-axis. Make sure you include your units. Now choose a sensible scale, so we're going from 1840 to 2020. Let's go up in 40s. Our mean yield goes from 2.4 up to 6.7. So I'm going to go 2, 3, 4, 5, 6, 7, and I'm using up my graph paper nicely. Now I'm ready to plot. So year 1840 we had 2.6. 1860 2.5. 1880 2.4. 1900 2.4. 1920 2.5. 1940 2.6. very similar. 1960, 2.8. 1980, 4.2. 2000, 5.3. And then a huge jump at 2020 at 6.7. And then it said use straight lines to join the points. In 1960, a dwarf variety replaced the old variety. Scientists compared the percentage yield in the two varieties. The percentage change in yield from 1840 to 1960 was just 0.06%. Calculate the percentage change in yield from 1960 to 2020. So use the graph. Let's read up 1960 to 2020 and then read off the y-axis to find these mean yields of wheat in tons. 6.7 minus 2.8 over the original times by 100. So you've got a mean increase of 139% over 60 years. But it wants it per year. Notice it's worth 3 marks. So do 139 divided by 60 to get 2.3%. Dwarf wheat has a shorter, thicker stem than the old variety, suggests why growing dwarf wheat is an advantage for farmers. So thicker stems mean less likely to be damaged by strong winds. So there's 1 mark. Because they're shorter, it means there's less energy to make them grow. And they're easier to harvest. There is a relationship between the colour of flower and pollination by insects. Design an investigation to find out if the colour of flower affects how attractive it is to pollinators. Include experimental details in your answer and write in full sentences. If you've been following me for a while, you'll know I like the variable layout here rather than columns. So what are we changing? I will choose a variety of flower colours, because after all that's what we're changing in this experiment, e.g. red, yellow, orange, pink. What about our dependent variables? So what are we measuring? Be really specific here. I will count the number of bees, because they're good insect pollinators, attracted to each colour of flower. Now the control variables, at least 3 or 4. What will we keep the same to make sure it's a fair test? We need to keep the species of flower the same. Type of pollinator. Generic things like temperature, time of day, weather. I imagine if it rains more, that will keep the bees away. Let's pick a time frame, 1 day. And you must repeat and calculate an average, and that will guarantee your full marks. So let's highlight the marking points. Different colours, that's a mark. Count the number of bees as a mark. In terms of our control variables, we've got a mark for species of flower, type of pollinator, temperature, so that's 3 further marks. We have a time frame mentioned, and we're repeating and calculating an average, so tons of marks.